Hello friends, assalamu alaikum. Today we will talk about the PCR. So PCR means polymerase chain reaction. Actually PCR means to amplify a particular piece of DNA, and amplification means to make several copies of a segment of DNA. So PCR is a technique used to make several copies of small fragments of DNA and RNA. So actually PCR is a process, or actually it is a technique which is used to amplify or to make the several copies of DNA or RNA fragments. So what are the components needed to perform PCR? So first we will need the desired DNA. Then we will need the DNA polymerase enzyme, and especially we will take the TOC polymerase enzyme because it is heat stable polymerase enzyme. Third one, we will need the 4 nucleotides: uh, thymine, guanine, cytosine, and adenine. Fourth one, we will need the buffer solution to adjust the pH during the reaction. Fifth one, we will need the primers, and primers are the short segment of DNA molecules which initiate the DNA elongation. Sixth one, we will need the thin wall tube, this is also called PCR tube. And seventh one, we will need the thermal cycler. This is a device used in PCR which regulate the temperature of the PCR during reaction. First we will need the DNA sample, then we will need the primers, and we will also need the nucleotides: cytosine, guanine, adenine, and thymine. We will also need the TOC polymerase enzyme. We will also need the buffer solution and PCR tube. So we will add all the material in the PCR tube, and we will place the PCR tube in the PCR machine. So what are the steps involved during PCR process? The first one is denaturing of DNA. So first we will denature the DNA. Denaturing of DNA means the separation of the strands of the DNA. At 95 degrees centigrade, the DNA strands separate from each other. You can see here the DNA strands are separated from each other. The second step is primer annealing process. The primers will be added to the strand of the DNA, and this reaction almost occur at 55 degrees centigrade. After annealing process, the third step is extension or DNA elongation process. At 72 degrees centigrade, with the help of TOC polymerase enzyme, new nucleotides will be added and two daughter DNA strand will be obtained. So first step is denaturing of DNA. At 95 degrees centigrade, DNA is denatured to two separated strands, the first strand and the second strand. The second step is primer annealing process. So at 40 to 65 degrees centigrade, the primer's annealing process occur, and the primer will be attached to the strand of the DNA. So second step is primer annealing process. So at 40 degrees centigrade to 65 degrees centigrade, the primers will bind to their complementary sequence on the single strand of DNA. So this is the first strand of DNA. So this is the 3' end. Here the hydroxyl group will be at 3 position, and this is called pio prime end. So here the hydroxyl group will be at pio position. Here the primer will be attached to the first strand of DNA, and elongation will occur. Further nucleotide will be added with the help of polymerase enzyme. This is called forward primer, and this is called reverse primer. Here elongation will occur in this position, and in the second strand elongation will occur in this position. So this is called a reverse primer, and this is called forward primer. So the third step is DNA extension. At 72 degrees centigrade, DNA polymerase extend the DNA chain by adding nucleotides to 3 prime ends of the primer. So this is the 3 end of the primers. Here new nucleotide will be added. So this is called DNA extension or DNA elongation process. So, and we will get 2 copies. These copies are called daughter DNAs. So at one cycle we will get 2 copies, and during second cycle we will get the 4 copies, and so on. So what are the application of PCR? So PCR is used in disease diagnosis, like to identify the disease causing organism, like hepatitis virus. During hepatitis we use PCR to identify the causative agent like hepatitis virus. And in the disease treatment, PCR also help in the disease treatment, like PCR is used to monitor the amount of HIV virus in the patient suffering from the AIDS, and this monitoring of the amount will help in drug therapy adjustment. So if the amount of the HIV virus is high, so we can use high dose of the drugs, and if the amount of the HIV virus is going down, so we can adjust and we can reduce the dose of the drug. In forensic, PCR is also used to detect the criminals. The forensic persons collect the sample from the crime area, then they extract the DNA from the sample, and then they use the PCR to make several copies of that DNA, and then they try to match that DNA with the suspected or suspicious person. The use of PCR in genetically modified food detection. Genetically modified food are the foods which are genetically modified using the genetic engineering to increase the production and to increase the taste and to increase the shelf life of the food. So in some countries PCR is used to check the exact quantity of the genetically modified food in a shipment. As we know that there are various advantages of the genetically modified food, but there are several disadvantages of the genetically modified food, like several diseases including immune suppression, including the insulin-less production, and including the infertility. So that's why some countries also use PCR to check the exact quantity of genetically modified food in a shipment. Use a PCR in paternity testing. During paternity testing, the PCR is used to find out the real parents of a child, and in archaeological study we also use the PCR. So this was all about the PCR.